Our next and final speaker of session two is Cara Hunter from Syracuse University, and I will hand it over to her.
All right, thank you so much. Let's see. I want to share my screen. Is this showing it in full screen?
It is, but it is also showing your notes at the bottom, so it is a little bit small.
Oh, hold on. Wonder how about this? Does it show the full?
That's perfect.
Okay, perfect. All right, so I'm Kara. I'm Kara Hunter from the Sucenic Lab. I'm a graduate student that's kind of been developing this pipeline for a little while now. And my area of research is to design disordered proteins and using this screening with a sequencing-based approach. So, as you may know, the Sucenic Lab is really interested in disordered proteins. So we are interested in finding out how we pare down the sequence to function paradigm. So disordered proteins have very low conservation, which makes studying them quite difficult. So we kind of um, are interested in investigating more of like the sequence chemistries to elucidate this molecular function. And to do that, if usually that involves doing thousands of different experiments to test thousands of different disordered proteins. But in my case, I wanted to figure out how to generate a library and make a massively paralleled assay to discover more of what this central dogma is of the primary structure of the sequence and how that begets molecular function. So there are some methods of generating IDP, disordered protein libraries. There's gene ontology terms where you can have a specific function that you're wanting to screen, or you can look at things specifically like with disordered proteins, with Disprot. These are databases that you can use to create these massive files. And we want to elucidate this hidden chemical grammar. So we use this Python package developed by our collaborators in the Whole House Lab called Goose. And this Python package actually specifies specific properties. So we take an amino acid sequence here and we generate variants with increased charge. So in this case, we increase the negative charge to positive charges. So like we have these lysines and arginines now. And whereas in another variant, we have more negative charges. So then we have this aspartic acid popping up more so than where we have the original variant. But this Python package isn't just limited by charge. We can also change the hydrophobicity. We can change the aromatics. And this basic parameterization of the IDP is what makes this Python package so powerful, because then in this big library experiment, we can have specific questions and hypotheses as to what chemical feature results to a given function. So we know that expressing disorder proteins is a prevalent mechanism of desiccation protection, as was so easily or eloquently said by our many other speakers today. So in Arabidopsis thaliana, in the seed, we have these biochemical protectants, which are found in leas co-expressing with small solutes like trehalose. In the water bear, the tardigrade, these are Cas proteins. And all of these just happen to be disordered. But in order to investigate them, we want to see what kind of um, sequence chemistries are interesting here. We have a little logo chart here of a Leia_4, and this just highlights how little homology there is within the Leia family itself, where you see the most abundant motif here, this 11-mer motif, is even then pretty weakly conserved. So we like using this library in a high throughput way to investigate what exactly makes these sequences tick, because we can't rely solely on homology. So with this experimental workflow, I synthesize this library on a chip. So each each DNA base pair is synthesized individually on this chip, and they're all pooled together. From there, I amplify these DNA base pairs, and I clone it with a linearized plasmid backbone and transform it into yeast. Yeast are really good at picking up DNA, so they actually recombine it in vivo. Then from there, I grow up this transformed library. I desiccate it in a humidity controlled chamber for 8 hours on filter paper. And then I rehydrate it, monitor its outgrowth. And before and after this desiccation, this selection, I send out these extracted plasmid libraries for sequencing. So before and after the selection, I extract this library, amplify the DNA, send that out. So this is basically just how I pellet the cells, extract it, amplify. And then when I confirm it on the gel, you need to make sure that it's all very specific, highly abundant in that one size fragment. So we assess this protection by sequencing. So what exact sequence is actually offering this protection and recovery? It's actually the output of this NGS service. So for example, we have a blue and red protein. So the blue protein and the red are both equally abundant before selection. Then after, you see that the red kind of overtakes this and dominates the culture. Next generation sequencing actually provides the read output. So it gives us blue has 4 counts, red has 4 counts. Then after the selection, we see that blue has 2, red has 6. So we would conclude that the blue sequence here is actually deleterious and the red is protective. And that's kind of the central methodology of my experiment. We can do this with my thousands of different proteins. So I have average reads before the selection between 3 biological repeats minimally, and the average reads after selection. This gives us a log-fold change, which is just calculated by the reads after, the average reads after divided by before, the log base 2, and the p-value, which is just a statistical p-test between these. So from here, I can generate what's known as a volcano plot. So the x-axis is the log2 fold change. Anything positive would mean that it's protective. So there's more of these sequences post-desiccation. And then anything on the negative side would mean that it's deleterious. So these tend to be worse than, say, a control. And the y-axis is the log 10 p-adjusted. That's the p-adjusted value here. Now I highlighted a specific subcategory of my library, which are the variants generated by that Python package I discussed earlier. And you can see kind of a distribution of all of those. And for example, protein C, we have a negative log-fold change, a slightly significant p-adjusted value, and that's how you see that it's slightly above that dashed line of significance here. Then we have, and I'll highlight, for example, an enriched sequence where it has more reads after the selection, and that's how it lines up with this volcano plot. So that's how we can kind of profile what sequences are significantly enriched and to what extent. Next, I kind of add this threshold here where anything beyond a value or an absolute value of 0.5 would mean that it's significantly enriched. So if you have a sequence that is only like 0.2 log fold change enrichment, that's not really significant enrichment. So we want to make sure that we are actually profiling signal that's very significant, I guess. So from here, we can explore these variants further and see how the log fold change or that log of viability change increases based on the specific parameters we're changing. So for example, we have a wild type sequence and we have a given like X number of variants just based on the changing of fraction of negative residues. So the the y-axis is the viability change now, while the x-axis is the specific sequence parameter. So right now, as you increase the fraction of negative charges, we actually see an increase in viability, which is really fascinating for the sequence, these variants. Whereas in another variant, we see that as we increase positive charges, that's actually deleterious. It has a negative of log of viability change. This is looking at these wild type sequences and their variants. There's not really a one size all fits answer. Between all of the different wild type sequences that we profiled within the library and all the different chemistry variations we've had, there's no specific dogma, I guess. So you have one sequence, one variant where the fraction of negative charges is really important. It's a really good predictor of protection. But in all these other variants that we've profiled, that's actually not a sequence chemistry that's important or that is a good predictor. For example, another one is the count of negative or neutrals. So there's never a one size fits all, and optimization depends on the sequence that you work with from the very beginning. So I guess that's time for me. Thank you for listening to my talk and all the collaborations I formed along the way with the Whole House Lab and the Boofy Lab. And I would like to thank my undergraduates too, my little team of people. And I'm ready to take any questions.
Thank you so much, Kara. That was great. We already have one question from Olivia. She said, great presentation. What is the percent humidity during the 8-hour desiccation? She also has another question. Was it one desiccation event or repeated cycles of desiccation and rehydration before sequencing?
Right. So we have it in a 9% humidity controlled chamber with sodium hydroxide pellets. And I just do one round of desiccation. So there's one event, dry it down for 8 hours, rehydrate it, and then upon outgrowth, that's when I sequence it for the after selection.
Awesome. Do you have a hypothesis on why negative charged amino acids correlates with increased recovery?
So I think charges are actually pretty important for Leia proteins in terms of interacting with water and kind of its environment. So maybe it has to do with water retention of some sort within the cell. But then again, I think this is a very nuanced answer because it depends on what kind of Leia family you're investigating. Maybe different Leia families have different mechanisms of protection. Who knows? So it just depends on what kind of wild type sequence you're looking at, I think.
Awesome. So the next question is, could you use your results of adaptive, don't know how to pronounce this, but deleterious, I believe, to
Yes.
further train Goose to directly predict desiccation tolerance? Thank you.
So I don't think we'll train Goose, but we'll definitely a machine learning algorithm of some sort. That would be really exciting to see if there's something that a machine learning algorithm can pick up in these sequences that we can't pick up by the human eye or by just looking at different chemistry analyses. So that would be really exciting if we can find a way to train an algorithm that would successfully predict basically the viability change and to then create new sequences that push the envelope of what's already protective or what's already deleterious.
Okay, and then we have one last question. Do you see any systematics regarding protein length?
Yeah, so I'm actually very limited on protein length with this library. I need to have everything pretty much the same exact length. But because of the on-chip synthesis, everything is limited to 90 amino acids maximally. But some of the proteins do have earlier stop codons. So, and then everything else is non-coding. We don't really see anything different in terms of length with those non-coding versus those longer sequences. And with our complete and non-coding controls, where it's just a stop codon alone, and then there's nothing else, it's just a non-coding, we see that those don't have significant enrichment whatsoever. So that's a good sign, meaning that, they shouldn't have significant enrichment because there's no sequence there to promote protection.
Awesome. Thank you so much.